Good morning, children. We have started with the second chapter, that is biological classification, in which we have started with kingdom Monera. We have studied in detail about the bacteria, its structure, the types of bacteria, then the mode of reproduction in bacteria. Then we have studied about uses of bacteria and harmful effects of bacteria. Today, we will start with the new topic, that is Archaebacteria. Archaebacteria, archae means ancient and bact means rod. So Archaebacteria are special since they live in some of the most harsh habitats, such as extreme salty areas called the halophiles, hot springs called thermoacidophiles, and marshy areas called methanogens. The characteristics of this domain are: they are most primitive prokaryotes. They are found in stressed environment conditions such as high salt concentration, Great Salt Lake, the Dead Sea, then edges of the ocean, hot sulfur springs, volcanic walls, gut of animals, etc. Their cell walls lack peptidoglycan. In most cases, the cell wall is composed of non-cellulosic polysaccharides and some proteins. In some members, cell walls may even be absent. This feature of having different kinds of cell walls is responsible for their survival in extreme conditions. Most of the Archaebacteria are chemoautotrophs. Now, types of Archaebacteria. Archaebacteria are of three different types. Methanogens. Number one is methanogen. They are strictly anaerobes. They live anaerobically in gut of several ruminants such as cows, buffaloes, goats, etc. These bacteria have been exploited for the production of methane, gobar gas, from the dung of these animals. These bacteria also help in fermentation of cellulose. They produced almost 65% of atmospheric methane. Examples of methanogens are Methanobacterium, Methanobacillus, Methanosarcina, and Methanococcus. The second type of Archaebacteria is halophiles. Halophiles are found in extremely saline environments like salt lakes, salt marshes, salt pans, salt solutions, etc. They are mostly anaerobic. They contain a chemical called halorhodopsin to pump in chlorides into the cell to prevent cellular dehydration. Halobacterium develops purple membrane having photoreceptor pigment, bacteriorhodopsin. In light, it acts as a proton pump and helps in synthesis of ATP. The formation of ATP is a survival mechanism under anaerobic conditions. Examples are Halobacterium and Halococcus. The third type of Archaebacteria are thermoacidophiles. This Archaebacteria can live in both extreme heat and acidic pH, that is around 2. Under anaerobic conditions, these organisms oxidize sulfur to sulfuric acid. Thermoacidophiles can survive in high temperature and low pH conditions because of the special features like spatial branched chain lipids in cell membranes that reduce cell fluidity, enzymes that can work at low pH, enzymes that are resistant to high temperature coagulation. Examples are sulfobolus, thermoplasma, and thermoproteus. Now, archaebacteria are very important. Archaebacteria can live in extreme conditions, so they are very useful. They are useful in modern biotechnology, generation of biogas, thermophilic enzymes, biosensors, restriction enzymes, etc. Now, let us move to the next topic, that is Eubacteria. Eubacteria are called true bacteria and are characterized by the presence of a rigid cell wall and is motile. They have flagellum. Let us study about the types of Eubacteria. Like cyanobacteria. Cyanobacteria, member of this group, have many characters similar to bacteria. These are also known as blue-green algae. The examples of cyanobacteria are nostoc, oscillatoria, spirulina, rivularia, anabaena, etc. They can survive in a wide variety of habitats such as hot springs, sea water, polluted water, etc. They are predominantly aquatic, but many species are known to be terrestrial also. This can grow even in minimal moisture conditions. The diagram which you can see in the background is this diagram which you can see in the background is the diagram of filamentous blue-green algae nostoc. Cyanobacteria have three forms. Number 1, unicellular as in Crococcus and Cyanococcus, colonial as in Microcystis and Gluocapsa, filamentous as in Nostoc, Oscillatoria and Anabaena. Cells of Eubacteria have a definite, firm and rigid cell wall surrounded by mucilaginous sheet. It is bilayered. The inner layer is made up of peptidoglycan. The cell wall is followed by plasma membrane made up of lipid and proteins. The membrane-bound structures like true mitochondria, endoplasmic reticulum, Golgi bodies, etc. are absent. The photosynthetic pigments present in the cell are located in broad sheet-like lamella called thylakoids. The pigments are chlorophyll A, beta-carotene, myxoxanthophyll, myxoxanthin, C-phycocyanin and C-phycoerythrin. The C-phycocyanin is blue in color while C-phycoerythrin is red. The former is present in more amount than later. This gives the cyanobacteria its characteristic blue-green color. The nucleolus is absent and nucleoid is not bounded by nuclear membrane. Some cyanobacteria like nostoc, Anabaena, Cytonema possess special type of cells called heterocysts to perform special functions. These are large cells with walls so thick that they are impermeable to oxygen. Heterocysts are the sites of nitrogen fixation. Cyanobacteria are mostly photoautotrophs. They contain chlorophyll A and other photosynthetic pigments. Now, reproduction. Cyanobacteria multiply asexually and vegetatively. Sexual reproduction does not occur. The types of multiplication are: number 1, binary fission occurs in unicellular forms. Fragmentation occurs in colonial and filamentous forms. The small segments formed are called hormogonia. Asexual reproduction involves formation of different structures like endospores. Endospores are the spores produced endogenously within a vegetative cell. Exospores, which are the exogenously produced spores. Akinets, these are thick-walled spores that also have food material stored in them. The diagram which is there on the screen shows a dividing bacteria which has a cell wall, cell membrane and DNA. Now, uses of cyanobacteria. Some cyanobacteria have the ability to fix atmospheric nitrogen. The green manuring by farmers is done on this basis to enrich the soil with nitrogenous fertilizers. Cyanobacteria like Anabaena, Tolyptorix, etc. help in prevention of soil erosion and its conservation. Spirulina is a protein-rich supplement for humans. It is a fast-growing cyanobacteria. It is also known as single-cell protein, SCP. Cyanobacteria like Anabaena and Olocera prevent mosquito larvae to grow in surroundings. There are certain harmful effects of cyanobacteria. Like cyanobacteria discolor the walls and roofs of buildings, monuments and statues. Oscillatoria causes asthma and gastrointestinal problems by releasing its toxins. Growth of oscillatoria in water bodies shows pollution by organic matter. Exclusive growth of cyanobacteria causes water blooms, which decreases oxygen level in water, causing death of aquatic animals. The next type of Archaebacteria are the Mycoplasma. Mycoplasma are organisms that completely lack a cell wall. They were discovered by Roux in the year 1898 in pleural fluid of cattle suffering from pleuropneumonia. The organisms are often called PPLOs, means pleuropneumonia-like organisms. The characteristics of mycoplasma are the smallest, zero point 0.1 to zero point 0.5 micrometer free living microorganisms that are so small that they can easily pass through bacteria proof filters. This occur saprotrophically in soil, sewage water, and in dead and decaying organic matter. Some of them also parasitize plants, animals, and human beings. Pathogenic mycoplasmas cause diseases in their host. They are unicellular with plasma membrane as the outermost boundary. The cell membrane is made up of proteins, lipids, and cholesterol. DNA is naked because of the absence of histones, and ribosomes of 70S type are present. Mycoplasmas possess heterotrophic nutrition. Example are Mycoplasma gallisepticum, Mycoplasma ledlavi. They cause pleuropneumonia in domestic animals, mycoplasmal urethritis in humans. They can survive without oxygen. So that is all for our today's class. In our next video, we will be starting with Kingdom Protista. I hope you all have understood this very well. Thank you.